All right, everyone. Hello and welcome to today's Love Data Week keynote address. Before I introduce our speaker, I'd just like to remind everyone that we still have sessions tomorrow, 3 great sessions for our Love Data Week tomorrow, as well as a couple more the following weeks. You can go to becker.wustl.edu to learn more about those and register. Also, big announcement, our WashU REDCap eConsent template is released and ready for use, and we have a couple webinars demonstrating how to use that template and adjoining SOP, which you can find information for those workshops either on our main Becker Library page or at becker.wustl.edu/classes/REDCap-workshops. Also, just a reminder that the NIH has a new data management and sharing plan that will begin January 25th, 2023, that will require all NIH grants to include a 2-page data management and sharing plan. If you would like more information about that, please email me at sorensenc@wustl.edu. But before all that, we have our keynote address today, and we are happy to welcome Dr. Russell Poldrack, who is the Albert Ray Lang Professor in the Department of Psychology and Director of Stanford Center for Open and Reproducible Science. His research uses neuroimaging to understand the brain systems underlying decision-making and executive function, and his lab is also engaged in the development of neuroinformatics tools to help improve the reproducibility and transparency of neuroscience, including the OpenNeuro.org and NeuroVault.org data sharing projects and the Cognitive Atlas Ontology. And the title of his talk today is Toward an Open Ecosystem for Neuroimaging Data Analysis and Sharing. And what a great topic that is as we prepare for the aforementioned NIH data management and sharing policy. And so with that, Dr. Poldrack, I am going to stop sharing my screen and I will pass it on to you.
All right. Well, thanks very much for the invitation. It's great to be here, and it looks like a really amazing event that y'all have set up. So I'm happy to be here to talk about some of the work that we've been doing over really the last decade in sort of developing an open ecosystem for open and reproducible research in the context of brain imaging. So, this probably goes without saying, but, we think that openness is a critical element for reproducibility. And that's why it's something that we focused on for so long. This is captured really well by a quote from Buckheit and Donahoe. David Donahoe is a statistician here at Stanford who's really been sort of on the reproducibility bandwagon since, before almost anybody. They said, an article about a computational result is advertising, not scholarship. The actual scholarship is the full software environment, code, and data that produced the result. And so we take inspiration from this quote and this concept in thinking about, what kind of tools and resources scientists need in order to be able to really do, reproducible work. So I'm going to talk about a number of different types of resources that we've been involved in building. One is a set of tools around open data, providing researchers with the ability to share and reuse, various types of data. The second is tools, open source tools that allow researchers to work with those data in an open and reproducible way. And all of this is sort of undergirded by work that we've done on open standards. In particular, I'll talk about a standard that we've developed along with a large community called the Brain Imaging Data Structure, or BIDS. So interestingly, in the functional MRI, so functional MRI is the tool that my lab works with most. And it's basically a way to look at brain activity using MRI. And interestingly, back around even before 2000, a group at Dartmouth, led by one of the founders of the field of cognitive neuroscience, got funding to put together this thing called the fMRI Data Center. And was sharing. In this case, they had about 110 data sets available. They this person who was running it was also involved in a number of journals and ended up basically getting both PNAS and the Journal of Cognitive Neuroscience to require researchers to upload their data to this database upon publication of their papers in those journals. And this, it's fair to say the field was not ready. And so we saw this sort of, erupted. This is a couple of different things that came out in Nature Neuroscience in 2000. And a lot of it was driven by, this is an excerpt from a letter that was sent around. This letter comes from a group of scientists who are publishing papers using fMRI to understand the links between brain and behavior. We're writing in reaction to the recent announcement of the creation of the fMRI data center. In the letter announcing the creation of the center, it was implied that leading journals in our field may require authors of all fMRI-related papers accepted for publication to submit all experimental data pertaining to their paper to the data center. We are particularly concerned with any journal's decision to require all authors of all fMRI-related papers accepted for publication to submit all experimental data pertaining to their paper to the data center. So clearly, in 2000, our field was not ready for data sharing. But if you fast forward, to today, I would say that data sharing, maybe it hasn't become the norm in the field of neuroimaging, but it's certainly becoming the norm. To the degree that, a couple of years ago, one of the big conferences that we have, the Organization for Human Brain Mapping, one of the well-known senior researchers in that field was overheard saying, OHBM has been taken over by the open science zealots. And interestingly, this person has now kind of become an open science zealot. So, we've seen in starting around 2009 with the publication of a really prominent paper by a large group of researchers that demonstrated the utility of basically aggregating data from many different labs to basically drive new discoveries. We've seen many different approaches to data sharing. They vary in one way, they vary in sort of the amount and the kind of data that are shared, right? With some projects sharing raw data, others sharing, highly processed data, others sharing sort of stuff in the middle. And so, these are some of the projects you may have heard of, particularly, given that you're at WashU, the Human Connectome Project, which was based at WashU, is probably one of the best examples of what we call a prospective data sharing study, where the data were collected with the direct intent of sharing the data basically, in batches as they were collected. And I think that's become, incredibly powerful. So, that study collected 1,200 data from 1,200 adults, shared all those data. And actually this 600 number is probably outdated by now. My guess is that it's at this point, at least 700 or 800 papers, that it's become a real workhorse data set for our field. There's a number of other projects that are ongoing. One really prominent one is the Adolescent Brain Cognitive Development Study, the ABCD study, which is following about 10,000 children over the course of a decade, repeatedly doing neuroimaging over time to try to look at the relationship between development, both cognitive development and brain development, and certain outcomes related to drug use. We've been focused on, what you might think of as the flip side of those studies, what we call retrospective data sharing, which is when, somebody does a lab does a study, they collect a data set, and when they go to publish the paper, they want to then publish the data set so that other researchers can either try to reproduce what they've done or extend it or mash those data together with others. And within the field over the last decade or so, we've developed what I think of as sort of an ecosystem for data sharing that varies along this dimension of kind of breadth versus depth. So on the one hand, you have this repository that I'm not going to say much about today, called neurosynth.org, which basically kind of scrapes the highly processed results from published papers. There's a particular format that people in our field publish the locations of brain activity in. And so Tali Arconi, who developed the site, basically came up with a way to scrape those out of the papers. And then, so we have data from about, I think it's 14,000 papers right now, and you can do online meta-analysis asking, a number of different questions. The data are everything I'm going to talk about is completely open. So Neurosynth, both the code for the platform and the entire database are all openly available. There's a Python package to do kind of work more directly with the data. On the other side is a project that I'm going to spend some time talking about called OpenNeuro, which we started developing. It started earlier as a project called OpenfMRI, and then in 2017 became OpenNeuro, kind of generalizing out a bit from just functional MRI data. And it's a project that you'll see has many fewer than 14,000 data sets, but it has much more, much deeper data because it has raw brain imaging data from each individual in the data set. And then in the middle, we have this project that I won't say much about either called NeuroVault, which is meant for sharing sort of the statistical results of a study, but in a more detailed way than are shared in the Neurosynth project. So, we really have a broad range of different tools. Now, you probably know this, right? That sharing data is not sufficient on its own. You need to share it in a way that makes it useful. And I think this is really well captured by this cartoon you could find on YouTube, the data sharing and management snafu in 3 short acts. This is it's kind of a blurb. It's the sad pandas trying to get some data from someone who published a paper in Science. And they say, I received the data, but when I opened it up, it was in hexadecimal. Yes, that's right. I cannot read hexadecimal. You asked for my data and I gave it to you. I've done what you asked. Later, is there a guide to the data anywhere? Yes, of course. It's the article that's published in Science. If you watch this, and you've been involved in trying to obtain data from others, it'll be sort of funny but painful. So we began back in 2015 an effort to basically develop a standard for organizing the data and the metadata of a brain imaging data set that has come to be called the brain imaging data structure, or BIDS. And it's actually been really successful. I'm going to talk a bit about what it is and why we think it's actually been successful. So we held an initial meeting in 2015 because we had, I'm going to tell you later about the OpenNeuro project. We had at that point obtained funding from OpenNeuro, and there was a challenge that we were having in a previous data sharing project, which was that people would give us data, and we had a kind of an in-house format that we would for organizing the data, and we would have to we have a curator who would sort of curate the data, put them into our format, and then run them through our analysis stream. And the only way we could really know if the data were properly formatted or not was if the analyses crashed. And that just wasn't a tenable procedure for us. And so we wanted to develop a standard, one that would be much broader than just our one lab, and 2, that could be automatically validated. And I'll come back to that in a bit. So we held a meeting initially in 2015. Later that year, we posted a draft standard to the BIDS website with a bunch of example data sets. We think that having examples for people is really important. And we had a period of open community feedback. We like to take as much feedback as we can from the community. And then later in 2016, we published this paper in Scientific Data that lays out the standard. So something interesting happened. So Chris Gorgolewski, who was a member of my lab, research scientist in my lab, he really kind of drove the instantiation of BIDS. He led that first meeting, led a number of subsequent meetings that we had at Stanford and Princeton. And then he left in 2018 to move to Google. And there were a lot of people who were really worried that this whole thing was going to fall apart. Because it's pretty common when a founder leaves a project that things fall apart. And what's instead happened is that it's become this amazing community project. So we now have community governance. We have a steering group that I'm a member of, was elected a couple of years ago. We have elections every year to kind of rotate people in. We have, at last check, 71 contributors on the GitHub page, which is where most of the action happens in terms of discussions of either extensions or changes to the specification. And then we currently have 6 maintainers, who are people some of whom are paid for by my group, some of whom are paid for by other groups, who work to basically help people bring ideas into the specification. And so it's we have community meetings every year at the Organization for Human Brain Mapping meeting, and regularly we'll have 50 to 100 people at those meetings. So we started with a few principles that were in part kind of driven by our experience with other projects. The first is that we thought that the main thing that we need to worry about is actually getting people to use this thing. Adoption is crucial. And so one of I think one of the key ideas that we had that's been really powerful is to keep the standard as close as possible to what people are already actually doing in their lab. And it's almost certainly not going to be, it's not going to be buzzword compliant. It's not going to be as technologically sophisticated as it might be. But what I've seen in a number of projects is that technology often overrides usability. You know, if you start telling people they have to use sort of complex file formats or complex technology stacks, you know, they just walk away. We also focused on engaging the community and making sure that we were doing whatever it took to listen to what people wanted out of the thing. The second is not reinventing the wheel. So whenever possible, we use existing standards. Sometimes we had to kind of slightly roll our own, but in general, as you'll see, we've worked with sort of common standards. The third, which is maybe the hardest, is being really focused on the 80-20 rule. Right, the idea that there's always going to be edge cases, and there's a set of people in any project like this who focus only on the edge cases that can sometimes be impossible to actually solve. What we said is we want to find the things that will let 80% of the scientists do the things they need to do, and not let the perfect be the enemy of the good. And we know that it's not going to be able to solve every problem for every person, but it'll solve enough problems for enough people to have value added for the community. So here's what the standard looks like. So when data come off of an MRI scanner, they look like this. They follow a standard file format that's been agreed upon by the manufacturers of MRI scanners called DICOM, which is a format that researchers never use. We almost always immediately convert our data into a different format called Nifty. This is these nii.gz files, and we put them together in ways that sort of make sense. So this is what a data set would sort of look like in an imaging lab. So we have, you know, in any imaging study, we would bring in a bunch of people, we call them participants. In this case, each of these, or subjects, we call them. So sub 01 is one person, sub 02 is another person, and so on. Within each of those subjects, you have different types of data. You have anatomical MRI, you have functional MRI, you might have diffusion weighted MRI. There's other types of imaging, but the data are sort of laid out such that you can kind of, you know, see what's where and kind of know what you're looking at, where, you know, as opposed to these sort of like just, you know, big strings of numbers. So in terms of like, you know, the file formats that we use in BIDS, we standardized on this thing called Nifty. Our field is actually really lucky that about 20 years ago now, a bunch of people came together and said, hey, we have all these different file formats that different software packages are using. We need to come up with a common file format for brain imaging data. And this is what became this file format called Nifty. Now has a number of extensions, GIFTY, SIFTY, but they all sort of follow the same pattern. And so we've standardized on that particular file format for BIDS, at least for MRI data. Whenever possible, we use just plain old tab delimited, tab separated value files, text files. So for example, this participants file, you know, we might have information about the age and the sex of the person. And then for things that require a little bit more structure, we store them into JSON files, which is pretty much every programming language has the ability to, to read and work with JSON. And so, for example, if we need like dictionary type, structures, we would store those in a JSON. So every image file has what we call a sidecar file, which is a JSON file that has a bunch of metadata, because the Nifty, one of the shortcomings of this Nifty format is that its metadata is pretty limited. So we store a lot more metadata in these JSON files. There's a question I'll come back to in a moment once I'm done sort of walking through this. Then the other thing that the standard comes with is a set of templates for the naming of both directories and files, right? And those allow sort of some flexibility in terms of, what the data types are, whether you have different sessions, for example. And then we have a fairly, complex file naming template where, many people think it's really bad practice, right, to embed metadata into the file names. And we also, store metadata in these JSON files, but researchers in general there's a certain set of bits of metadata that they really like to have in the file name because it makes it really easy to know what they're looking at. And so we made the decision to go ahead and embed those into the file name. So let me answer Akshay's question. With the 80-20 rule, how do you handle situations where specific edge cases might provide high amount of value due to its unique nature? How do you prioritize? I.e. more papers doesn't necessarily mean more value. I mean, in the end, it really comes down to a community decision, right? You know, that if somebody has a situation that isn't being handled that they think is important enough to be handled, what they do is they go to GitHub, they post an issue, and then they and the maintainers and the rest of the community have that discussion. And it certainly is the case that, you know, things have been added. There's also, as I'm going to show you, mechanisms that are sort of inspired by the way that it works in open source software to actually extend to new data types. And they'll allow, like, you know, completely new groups to come along and develop an extension that can then be incorporated into the main standard. I'll say more about that in a bit. Another question from Xian Young: when you convert file names coming out of MRI on your own, are they linked? And also, does the original raw data have any metadata that has information that needs to be de-identified? Both good questions. So there's a number of, I mean, there's no one way that people convert data from kind of, you know, their original data into BIDs. There's a number of different tools that people use. And so, you know, the answer to that first question really depends on exactly what tool you're using, you know, how that's going to end up being treated. Actually the second question about, you know, de-identification also is sort of, you know, somewhat site specific. I mean, in general, the BID specification has, you know, details regarding, for example, you don't put in specific dates of data acquisition. You don't put in individual names. You know, things need to be sort of numbered in a way that doesn't have anybody's, you know, individual name. So it's meant to be a de-identified format, but certain, you know, it is the case that, you know, if you, for example, just convert your DICOM header into a JSON file, it might contain identifiable information. And so it's up to the researcher to ensure that that's not the case. All right. So as I mentioned before, you know, one of the challenges is you really want people to easily be able to know whether their data are compliant with the standard. And part of the motivation for this was, as I said before, you know, we were having trouble keeping up with the volume of curation that was required to, you know, to get data into the kind of the predecessor of OpenNeuro called OpenFMRI. And we wanted to move to an automated data ingestion process where in some sense we're pushing curation back onto the users. And so we developed a validator. This is a JavaScript validator that can run in the browser. It can also run on the command line that basically very quickly looks at a data set. It looks at the metadata. It doesn't actually load the image files and tells you whether or not it's a valid BIDS data set. And if it isn't, it gives you information about, you know, why and what you might do to fix it. And so that's really helpful for people to, you know, to be able to easily look and see whether their, you know, whether their data set meets the standard before it's ever uploaded, because it can just be done in the browser on your own machine. There's a growing amount of tooling around BIDS. One of the, I think the most useful and important tools is this thing called PyBids, which is a Python package for working with BIDS data sets, developed by Tal Yarkoni, who moved to Twitter about a year ago, I guess, but, you know, was a really important contributor to our field. And what this lets you do is, you know, read in a data set, which is just a directory on a machine. And then you can say, you know, you can do various things like querying for subjects, finding, you know, what type of data a particular subject has, finding, you know, metadata for a particular image file, you know, retrieving it from the JSON. So, you know, a really kind of important tool. I mentioned that, you know, the original standard was really focused on several specific kinds of MRI: structural, functional, and diffusion MRI. But we also took inspiration, particularly from the Python world, and thinking about, you know, developing a mechanism for extension protocols. We call them BEPs, BIDS extension protocols. And there have subsequently been a number of BEPs that have been approved by the community for new types of imaging modalities, magnetoencephalography, various types of EEG, other types of MRI, like quantitative MRI. Recently, we had positron emission tomography, and then just very recently, there's a new extension for microscopy data. There's also a lot of work going on around different data types beyond raw data. And we had funding from the Brain Initiative a couple of years ago to start some of this, which has really been focused on moving to process data. So for example, when you run data through a pre-processing tool, getting data out of that follow a standard that somebody else can easily reuse. Things like connectivity matrices, statistical and computational models, image transformations, lots of different things that researchers use in their analysis all the time that we want to be able to share in a way that's reusable. So BIDS has been really, sort of strongly adopted. It was endorsed. It was actually the first community standard that was endorsed by this INCF, International Neuroinformatics Coordinating Facility. And basically they went through a review of the standard and basically said, hey, this is legit. It's been adopted by a bunch of different projects, and now there's I have 40,000. The number is almost certainly way bigger than that. There's a lot of anonymized data from people formatted in BIDS that are available either publicly or through data use agreements. Now, one of the things that we wanted to do is sort of in terms of this idea of prioritizing adoption is giving people an incentive to format their data in BIDS. We didn't want it to just be like medicine that people think they have to take because it's the right thing to do. We wanted it to be the case that if you put your data in BIDS, you're actually going to get value added. And so Chris Gorylewski developed this idea of what we call a BIDS app. And this is a containerized software application that can basically look at a data set, a BIDS data set, and immediately know how to run some particular software package on that data set. So some of the major software packages, for example, there's a package called FreeSurfer that's really commonly used that does structural MRI processing. So there's a BIDS app that basically, if you have a data set that's in BIDS, you just fire up Docker and point it at point the FreeSurfer container at your data set. And it will go and run that thing without you having to reformat your data at all, and also without having to install FreeSurfer, because all the BIDS apps are containerized using Docker. This also achieves reproducibility aims because those containers encapsulate a specific version of software and a specific version of all the dependencies of that software. So that when you rerun it, modulo weird random number stuff or floating point things, you should basically get the same answers. So we've developed a couple of these BIDS apps in my group. The first one I'll mention is called MRIQC, which was developed by Oscar Esteban, who's now in Lausanne, Switzerland. One of the challenges in MRI research for a while has been getting sort of really usable quality assurance metrics on a data set. And so what MRIQC does is it basically takes a BIDS data set, loads it in, and then computes a number of image quality metrics on both the structural and functional MRI data, and provides reports that allow you to look at the group, and then also to sort of dig in to look at data from any individuals who might be, for example, outliers on one of these image quality metrics. One of the things that we do here is to actually feed those metrics with a little bit of metadata back into a central database that's run by our friends at NIMH, where you can opt out of it, but if by default, the metrics and a little bit of metadata about the images are sent back. And so we now have, this is again an old number, that's well over 300,000 unique fMRI scans at this point. It allows you to ask questions, right? So for example, is the temporal SNR lower for higher multiband factors, right? Which will make sense if you're an imaging person. The point here is that there's a bunch of data that we can get by working in the open. These data are also in the open. Anybody can access them via this site at NIMH. And so it enables people to go off and do lots of their own interesting analyses. The other tool that we've developed, which is a BIDS app, is this thing called fMRIPrep. So when we do an fMRI study, we get a number of different MRI images, and then there's a ton of pre-processing that has to happen before we can ever do any sort of statistical analyses on the data. And so the way that it has generally worked is that, any particular lab will use some particular software package. There's several commonly used software packages in our field, and each lab would say, I'm going to use FSL, which is one of those packages. And they would do all their pre-processing in FSL. They would usually develop some scripts. The first postdoc in the lab who started doing fMRI would write some scripts, and then for 10 years, people would kind of keep hacking on those same scripts and trying to become sort of like a big, kind of Jenga pile of code, and is really not very reproducible. So we wanted to try to address that by building a really robust pre-processing pipeline that tries to basically use what we think of as sort of the best of breed tools for whatever it's trying to do. Rather than saying we're going to use FSL for every processing step, we're going to take different packages that can do things differently, and we're going to try to take advantage of that, of doing it sort of in the best way that we know how. And so one of the things that we try to do, the problem with a tool, or one problem with a package like this, is it's an incredibly powerful black box, right? People put data in, they get stuff out, and the danger is that they won't actually know what's happening on the inside, and they won't pay attention to, to making sure that things actually worked right. So what we do is we try to provide details about how things are working, but also provide really usable visualizations. So for example, this is a visualization that allows the researcher to very easily check the registration of two different types of data by basically animating a shift back and forth between the two data types with a cortical surface overlaid on that. So you can, if something has gone wrong, it will very quickly pop out at you in this type of a visualization. And so we try to basically, incentivize people to look at the places where it's really important to look by making it really easy for them to do so. We have a telemetry system set up within fMRIPrep. Again, you can opt out of it, but if you don't, whenever somebody runs a job, it sends a little ping to our telemetry server basically saying, hey, somebody ran fMRIPrep. Either it ran successfully or it crashed, and here's a little bit of information about the what module crashed, without actually sending any of the data or metadata. This is the data from the end of 2020 to 2021, so quite from about a year ago. And there we were seeing on the order of 2,500 to 7,500 runs per week. Now I unfortunately couldn't get a plot of the newest metadata, but now it's apparently like pretty consistently over 10,000 runs a week, successful runs. And we take advantage of this to sort of keep our eyes on what errors are popping up. So when we put out a new version, if it's crashing for a lot of people around a particular module, we'll see that pretty quickly. So now I want to talk about the platform that we've developed, which is one of several platforms in our field for the sharing of brain imaging data. And OpenNeuro, I think, has been successful because of the engineering that we've done to try to make it as easy to use as possible, and because of BIDs. So this is basically a free and open platform for sharing any type of data that's compliant with the BID standard. Well, except for microscopy. We're not going to share microscopy because those images are so big, and that's a kind of a different subset of the community. But anything that is kind of like macroscopic brain imaging, we share those data. So if you go to OpenNeuro, even without logging in, you can see these sort of public data sets and poke around. We actually we just implemented recently a new sort of interface and search procedure that actually works quite a bit better than the old one did. But basically, if you go to one of these, let's say you want to go to this top, they're sorted by views. This one's had 1.9 million views, 1,400 downloads. If we go into that particular data set, it has some metadata like the authors, a bunch of information, the license, the DOI. I'll say more about those. And then you can actually browse the data. One thing to note is that, you see over here on the left, there's these different versions. Before sharing anything publicly, a data set has to be basically snapshotted to create a specific immutable version of that data set. And it can be changed later. Sorry, you can change the data set and make a new snapshot later, but that particular snapshot will never change. And that allows us, again, to have much better reproducibility over analyses, because you know exactly what version of the data were used. And so you'll see, this tells us something about the BIDS validation. This data set is valid. Occasionally, a data set can become invalid over time if the BIDS standard changes. That doesn't happen too often, but it has happened on occasion. If you click the upload button, you want to upload a data set up here. The second thing that happens after you select your files is it goes through validation. And in this case, it says there's some warnings. You don't have to fix them. And it would show you those warnings and also link you to the BID specification. I mentioned this, all data sets are versioned. And if you want to download the data, there's a number of different mechanisms. For small data sets, you can just click the download button and it'll download through the browser. That tends to fail for really large data sets if we're talking about like hundreds of gigabytes. And so we generally recommend that people either use, because all the data all live on AWS. It's actually part of the Amazon Public Datasets project. So they support the storage and bandwidth of all these data for us. And so you can easily grab this without any sort of authentication. You can grab the data from OpenNeuro. We also have a node command line interface. And then there's another project that we rely on for our kind of back-end storage called DataLad, which builds on top of something called Git Annex, which is like Git for large files. And that also provides another way to grab those data. You'll see that we provide a DOI, which includes the accession number and the version number in it. And so, you can stick that in your paper. That's probably, I think the primary use case for people using OpenNeuro is because they have a paper that they're publishing and they need a DOI for their data set. We've recently also implemented the ability to generate basically anonymous reviewer link. So if you have a paper you're submitting and you want to allow reviewers to see the data, even if they're not yet made publicly accessible, because any data set, but you basically can be made like a private draft, and then you can publish it later. You can share private drafts with reviewers prior to making them public. This is just an example of one particular data descriptor that used OpenNeuro, stored their data, took advantage of that DOI. So we work under a CC0 license by default, which isn't, I know it's not a license, it's a public domain dedication, but we'll call it a license for the moment. And I think we have a paper that just came out about kind of the landscape of data sharing in neuroimaging. It came out this is with Annie Joie, came out in Human Brain Mapping just a couple weeks ago. And when you look across all the different projects that share data, we're definitely at I don't know if we're at the left or the right end of that distribution, but we certainly share data more openly than most projects. There's really no intellectual property restrictions because the data are basically put into the public domain. People can do whatever they want with them. No requirements for co-authorship, no prior restraint on any hypotheses or analyses. So the one thing this requires is that you really want your subjects to know that their data are going to be shared openly. And so a group in our field has developed this thing called the Open Brain Consent, which provides kind of template consent language for researchers who want to sort of make sure that their subjects are being really informed as to the nature of sharing that's going to happen. It also requires anonymization, and importantly, we need to remove facial structure. This is an example of one of the defacing processes. There's debate right now as to whether this really, eliminates the ability to identify people. There's some evidence that, things that remain after defacing, like the brow and maybe the head shape, can still, allow some degree of de-identification. And this is something that we're watching. But, working with our privacy office, they're not yet concerned that we can't say that removing the facial structure, along with all the other HIP identifiers, makes it de-identified. We're also working, because sometimes people will either accidentally or sort of, just negligently upload a data set that still has facial structure. Right now, we have a curator who goes through each new data set and manually checks for faces. And if they're there, they will take the data set down and flag that to the data set owner. We have a tool that we're developing that hasn't been implemented on OpenNeuro yet. We had a paper recently, or a conference paper on it, that basically it's a neural network tool to automatically detect the presence of facial structure. So we're hoping that will help. Ultimately, if we could run that in the browser, that would help, sort of on the upload side, identifying faces before it ever gets uploaded. So we published a paper just a few months ago in eLife that sort of laid out the project. And so if you're interested in learning more, you can certainly have a look there. This shows the growth of both number of subjects and number of data sets over time. So you can see it's been a pretty steady growth since 2018. We actually went live a little bit before that, but in 2018 we had to basically change out our backend infrastructure, and so we only have the data back to that point. It's a very diverse database, in the sense there's different types of imaging data. It's heavily skewed towards anatomical and functional MRI, but there's a growing amount of EEG data, some PET data, even some intracranial EEG data. Most of the data are from humans, but there's a growing number of non-human data sets from mouse, rat, non-human primate, dog, and pig. Then if you look at the kind of range of either the number of participants in the study and the number of sessions per participant, you can see there's a big range, and we kind of cover that whole gamut, along with the number of for data that are looking at different cognitive tasks. You see that there's a big range of the number of tasks that are being measured. So it's a really highly diverse data set. Obviously, it's not the data set, the database isn't particularly useful if people aren't reusing the data. So unfortunately, there's no great way to kind of automatically identify reuses. So we had to have a curator kind of dig through the literature, search for, people's mentions of OpenNeuro, and go through and sort of, find all the reuses. And this is a plot this is only for the first half of 2021. So obviously it's going to be much higher if we haven't gone back and finished it for the rest of 2021. But the point is that the number over the last few years, the number of published and preprint reuses has gone way up. So we think that our work is paying off. So what do we think the keys to success have been for data sharing in our field? I talked to people from a lot of other fields, and we see that they're challenging to get people in their field to even think about sharing data. I think we have some built-in advantages, which are that the data are digital from end to end, right? At least most of them, we get the data off the MRI scanner in digital format, we transform them. So, unlike other fields where people may be, sort of taking notes by hand or working with photographs and things like that, we're digital from the beginning. We have a set of standardized file formats and data standards. So we have BIDS, we have NIFTY. What this really does is makes it so that anybody can take a BIDS data set, and sometimes there's going to be information, that metadata that one needs that aren't there. But for the most part, the data sets are usable by almost anybody. And that also really reduces the burden of curation and preparation. So let me address the question that's in the Q&A. To track reuse of data shared, is it possible to add a tracking number into metadata? The challenge really is that, there's a DOI that is a digital object identifier that's provided for every data set. In principle, if somebody reuses the data, they should put that DOI into their paper. And then there is a project called DataCite that will then track those DOIs. The problem is that many researchers don't properly cite the data set in their paper. They'll often maybe say the data from OpenNeuro. Sometimes they give the accession number. Sometimes they give the DOI. If they gave the DOI each time, it would be trivial. And that would be, pretty much, then we could use DataCite and see it. So this isn't so much a problem on our end, it's a problem on the end of people writing papers, properly citing. And we tell people how to do it, but people don't always listen. I think another key has been, demonstrated scientific utility, that it's not, again, data sharing isn't just happening, because it's good for the world and it's like, taking your medicine. But there's a lot of success stories. There are people, there's a growing number of people who've built careers out of analyzing only shared data. And we can talk about whether that's a good thing or not, but it certainly is the case that, that it's the scientific utility of shared data is really clear. So what have we learned? Well, I think that we've learned first that community buy-in is essential. If you put a mandate in place before the community is ready, it can backfire unless, obviously this has happened in certain areas like in genomics, where a bunch of really powerful funding agencies and journals came together and said, we're going to require everybody to share their data. We didn't have that, right? And so there was early kind of, backlash. But I think it's grown over time, because the community is bought in. And I think an important part of that has been that the people who are advocating sharing, like me, have largely been members of the community. They're not people who are just like in the business of data sharing. They're people who are in the business of neuroscience, but they want to provide the ability for people to share their data. And we, in Silicon Valley, there's this term dog fooding, right? Basically, the idea is you need to use your own tools and, do the things that you like, basically, if you're you need to walk your talk in a sense, right? And so I think that's been, there's not just our project, a number of other projects where, researchers have been at the forefront of sort of showing how data sharing can work and actually make science better. I think another lesson was that, keeping it as simple and as close to standard practice as possible is really powerful. This is an image from an earlier project, this thing called the Biomedical Informatics Research Network, or the BIRN, which tried to develop a sort of a data sharing network. To be part of that network, you had to buy a whole rack, they call the BIRN rack, with all these different systems on it that would implement a storage resource broker, and I don't even know what the MCAT is, and those would talk to the portal mediator. And you can start to see just how complex this type of architecture is. And it was really technologically cool, and it kind of failed because it was over-engineered. And so my rule of thumb is if there's more than 2 novel acronyms, then you have to be worried about whether researchers are actually going to sort of get their head around it. Okay, so another question, does the repository have a way to check how many people download the shared data? If yes, can you count the data download as reuse, or does it have to be published to count as reuse? So yeah, we do have statistics on downloads. I should have put them in here. I don't have them immediately on my head, but we've given out terabytes of data even in just the last year. The challenge is you don't know what people are doing with it, right? Sometimes people download it and then just play around with it. Other times they're going to use it for science and publish a paper. So we can't, they might use it for training. There's a lot of reasons people might download the data and never actually publish it. And so we can't just look at download statistics in order to kind of quantify reuse. Finally, I think that, our adherence to the Pareto principle, right, the 80-20 rule, I think has really paid off, right? Not letting the perfect be the enemy of the good, not letting edge cases derail us, I think has really, shown itself to be the right strategy. And it's something that we, in our steering group, everybody there continually, whenever somebody comes forward and wants to propose something new, they continually, sort of keep that philosophy in mind. I'll come to Avi's question in a moment. So, I think that in some ways, this image here is meant to reflect the fact that we sort of live in a bubble, right? That we've built this actually really amazing ecosystem of open open tools, open data. And the reason that that's worked is really all about infrastructure. I think software infrastructure, web infrastructure, but it's also about community engagement. The fact that that we have a really open community, and people come together to sort of work on this stuff. But we continually need to worry about not outstepping the practice of individual researchers, because we need to keep the tools as close as possible to what they're doing. So I just want to thank everybody who's been involved in this, particularly Chris Gorolevsky, who really sort of started the BIDS project, and he and I, really sort of brainstormed OpenNeuro. Squishy Media is a company that had been involved for several years in actually developing OpenNeuro. They just went out of business, unfortunately, but they still played a major role. And obviously, a bunch of other people have given us money. We're currently, funded by the Brain Initiative as one of their, OpenNeuro is funded as one of their official data archives. So with that, I'm going to stop, and I will address Avi's question in the chat. A multi-institutional group specified that they will share their data, their raw data, in their funded NIH grant. All attempts to access the data were stonewalled. Thoughts? I guess I'm not exactly clear on who the stonewalling. Okay, so this must be the stonewalling by the researchers who are basically unwilling to give up their data. This happens, and unfortunately, there's not great. There's not great, teeth behind a lot of at least the previous policies for data sharing. I mean, I think that ultimately, that we now, we just put in a progress report for an NIH grant, and you have to show them for any publication that it has been put into the PubMed Central archive, right? And if you don't, they kick your progress report back, and you have to email with them and convince them that you did. And I think we should have the same thing for data. I think when you submit your progress report, if you have data that that you said you're gonna, for example, if you have a paper that you've published, you need to share the data that go with that paper, and that should be tracked just like we track publication. But yeah, and I don't actually know whether the new NIH data sharing and management requirements, whether they will give more teeth to NIH. I haven't read them in enough detail to know that. It'll be interesting to see.
Based on what we're reading, that's the plan. But yeah, we'll see when it gets implemented. Marcy has a question in the chat as well, and I'll also just let everybody know at this point, if you'd like to, you can continue typing questions into the Q&A, or if you'd like to be unmuted to ask your question, feel free to do that as well.
Okay. So yeah, I think Marcy's question is a great one. The very early days of infrastructure design and development can be very overwhelming to some to begin down a similar road. Do you have any advice? How did you build the team? Do you have any institutional IT help? We've generally not relied on our institution for IT help for the most part. We got funding to do the work, and so I do have several software developers in-house. We're fortunate enough to have funding for these projects that can support that. But most of the actual work on developing the OpenNeuro platform, for example, we hired out to a company, in part because there's when you want to build a website, like a big web database like this, there's a lot of different things that you need. You need somebody doing user interface design, user experience. You need somebody doing backend. You need somebody doing cloud deployment. And there's no way I can hire an expert in each of those things. The nice thing about hiring a contractors are expensive, but the thing you can get from them is 10% of a person doing UX, 10% of a person doing cloud deployment. And so you can kind of get all the people you need in one. The other thing I would say is I think we've worked by sort of an agile philosophy, which is that, we want to basically get something out. We want to have a product that is, we have another of our mantras is minimum viable product, right? What's the thing that we can get out that will do something? It's not going to do everything we want to do, but it'll do something. And then once it's working, we can go and add the next thing we want to do. Because the problem is, it's often, there's this temptation to kind of design this really amazing system and then spend years building that thing. Only to realize that it doesn't actually work in practice, or that people aren't interested in it. And so we've tried to work by this kind of this agile philosophy of, just getting things out the door, even if they can't do everything people want. And I have to say, building an IT team is hard, right? Finding people, hiring people is hard. We're lucky that it's easy for us to hire people remotely. So actually all of the software developers in my group live elsewhere. One is in Austin, one's in Portland, one's in Boston, one's in New Hampshire. And that's worked, so we were very well set up for the pandemic and moving to Zoom because we'd already been having our IT meetings on Zoom for a couple of years. But that's an enduring challenge for us. And we've been lucky to get some really good people so far, but it's definitely tough. So I can address Shuhao's question. Any open source machine learning tasks, benchmarks for OpenNeuro bids? Not that I know of yet. I think it's a really interesting question. I'm kind of surprised nobody's done it. And one of the things that Chris Gorgolewski really wanted to do before he left was basically, getting a bunch of data from OpenNeuro into the shape to put, say, on Kaggle into put it into the formats that, machine learning researchers could kind of work with, more easily. And that hasn't, really happened. So I'd love to see that, but it's not really there yet.
Right. I've been letting the audience go. I've got my list, so I'm going to be selective. So important question here. Was there any specific factors that led your anonymous researcher who had the comment about the zealots to become a zealot? Was there some breakthrough, or was it just over time, kind of a slow?
That's a good question. I don't know what the causal chain was that led to that. I'll say what I've seen in other domains. What I have seen is that graduate students have incredible power over their PIs. If they're always saying, "Hey, we want to share our data. Hey, we want to pre-register. Hey, we want to do open source." Blah blah blah. At some point, the PI just sort of succumbs, I think.
Yeah, that's great. Okay, and then so you've talked about your funding for these projects. We have a lot of a lot of researchers who are concerned about the funding, the cost of data sharing beyond the life of the award, and data management costs beyond the life of the award. The new policy requires that funds be used during the life of the award. Do you have any advice as far as other mechanisms that people could look into if they have a giant amount of data they need to store, and it's going to be a large cost?
I mean, it depends on what kind of costs one is thinking of. I think if it's about like infrastructure costs, I think there's enough archives out there that if people can get their data in while they still have money to hire people to do that, then I think, NIH is sort of committed to supporting those things. The challenge is really like, right, you're collecting data up to almost the end of the project, you're writing your papers, and suddenly you don't have a bunch of money to pay people to spend their time getting stuff in. So I think that really doing it and being more proactive is the right way to go. One is, like, if you're using bids, then it's very little work to actually get your data ready to share. They're basically sharing ready, because bids is the format that they'll need to be in for you to share them. And so it sort of kills two birds with one stone. I also like to use this idea, right, that sharing data isn't just about sharing with other people, right? When you want to come back and look at a data set a year from now, you're effectively a different person.
Exactly.
And so the work that goes into curating the data set and, for example, getting it into bids is equally important for you down the road, even if you never share it with anybody else. And so I think that from my standpoint, the only way to really address this issue of, like, having the resources for sharing is being proactive and making sure that as you're collecting the data, they're being collected and stored in a way that they're basically ready to be shared.
Yeah, it goes right along with our Love Data Week, the quote that metadata is a love letter to your future self.
Exactly.
You're the one who's most likely to reuse that data. So Dr. Poldrack, thank you so much. We are at time. If anybody has further questions, feel free to reach out to me. This was incredible. Thank you so much.
Sure, it's my pleasure. Thanks, everybody.